Okay, I'm happy to introduce the speakers from our second Phage II course, where we look at the bioinformatics of the genomes that were isolated by students in our class. So Rachel Martindale and Emma Topping are going to tell you about this really interesting thing they found as they compared the genomes of different bacteriophages. Okay, go ahead, you guys.
Can everyone hear me?
You can.
Okay, cool. So hi, I'm Rachel Martindale. I was in Phage Lab 2 this semester, where we analyzed the genomes of phages that were isolated in Phage Lab 1. So my partner Emma and I focused our project on this really interesting pattern of repetitive sequences in cluster Q mycobacteriophages. So those repetitive sequences are those red X's on this picture. So there's a few reasons why we analyze phage genomes. One reason is that by looking at phage genomes, we can understand some of the complex relationships between phages and their bacterial hosts, and it can also give us some insight into the evolution of phages. But more importantly, by analyzing phage genomes, we can ensure FDA approval for phage therapy against antibiotic-resistant bacteria. So by looking at phage genomes, we can make sure that there's no integrase gene. We want the phages to kill the host instead of integrating their genome. And we can also make sure that there aren't toxins, something that's dangerous to humans, or that there are genes that could be beneficial to bacteria. So the main program that we used to compare our phage genomes was PhamRater. So PhamRater groups phages into clusters if they have sequence similarity across 50% of their genome, and into subclusters if they have nucleotide sequence similarity over 75% of the genome. And then another thing that PhamRater can show us is that phage genomes exhibit synteny. So a lot of the time genes will happen in the same order. So this is what a PhamRater map looks like. So on the top lane is one phage genome, and then it's compared on the bottom to the second phage genome. So each box that you see represents one gene, and the colors in between indicate the sequence similarity. So purple means that the sequences in this region are identical, red means that there's significant sequence similarity, and white means that there's no similarity. And you can see the synteny in this PhamRater map too. So in both of these phages, there's a tail assembly chaperone, then the tape measure protein, and then the minor tail proteins. So you can see that conservation of gene order. So phage genomes are also highly mosaic. When I say mosaicism, I mean that phage genomes have really patchy sequence similarity when you compare them. So you can see that in this PhamRater map, where there's some parts that are purple, so they're identical. Some parts are white, so there's no similarity between phages in the same cluster, and then the other varying levels of sequence similarity. And one reason for this is that phages can undergo horizontal gene transfer with their hosts. So for example, when a phage integrates their DNA into bacterial DNA, once they excise their own genetic information, sometimes they'll take a bit of the bacterial chromosome with them. So we focused our project on cluster Q mycobacteriophages. There are 20 members, one of which, Galancho, was actually isolated at CU Boulder. And cluster Q phages are normally temperate, so they can integrate their DNA into bacterial DNA. So when we look at their genomes, we expect to see an integrase, a repressor, and an excised protein. So when we were looking at cluster Q phages on PhamRater, we were noticing these really interesting sequence repeats. So you see these red X's occurring between all cluster Q phages. So first, it happens between genes 19 and 20, so an unidentified gene and the big, long tape measure protein. These repetitive sequences also happen within two minor tail proteins. And then there's also one X that happens in the non-coding region upstream of the integrase gene. So we looked at some other PhamRater maps of different clusters and saw that these repetitive sequences aren't all that uncommon when you're looking at minor tail proteins and tape measure proteins. But when you compare cluster Q phages with the clusters I showed you earlier, you see that you're not seeing those repetitive sequences between these different clusters. So they're not the same sequences happening. So then what we did next was we did a blast analysis of the cluster Q phage AMI-MEC So BLAST will compare a sequence to a really big database of sequences and see the statistical significance between sequence similarity. So what we did was we blasted that region of AMY-MEX genome that has the minor tail proteins in the integrase gene. So as expected, we got a lot of other cluster Q blast hits, but this little pink region of alignments shows that there's a region of AMY-MEX genome that could have come from mycobacterial species. And when we narrowed down exactly where those nucleotides were, we saw that it actually corresponds exactly to that non-coding region upstream of the integrase. So this sort of suggested to us that this might be the site of recombination when cluster Q phages integrate their DNA into bacterial DNA.
Okay, I'm gonna teach you guys how to read something called a dot plot really quickly. So we're gonna look at a really normal dot plot first, which is this cluster Q major capsid protein. Basically what a dot plot does, it's like BLAST in that it compares two genomes. Dot plots compare the genome on the left to the genome on the right. So if you look at AMY-MEX to AMY-MEX, of course they're the same. And then one square over, you'll see AMY-MEX to DAGAL. And so when we look at dot plot comparisons, what we're looking for to see perfect similarity is sort of this perfect diagonal line with no missing parts or no extra lines. So this is an example of that. And then if we look at a different dot plot comparison of one of these places where we are seeing sequence inversions and sequence repeats, we can see that it's a little bit different. And it has some extra lines next to the diagonal, which tells us that there are some sequence repeats in there. And then it also has this sort of red perpendicular line. And to us, that tells us that that's an inversion. So that means that when we're reading this DNA backwards, it matches up with the regular DNA. And if we look at one more comparison, it kind of shows that those inversions don't always happen when we see that red X. So here you can see that on Famarator, we do have the red X indicating some sequence reoccurrences. And then on the right, you can see that there aren't any of those red perpendicular lines. So we're not considering those an inversion. And an inversion mutation happens mostly because phage are really highly mutable and they can evolve really quickly. Some people think that the phage encoded recombinases may be more effective at rejoining broken DNA to become like a greater whole, and that this means that they have some differences from other organisms' DNA. And an inversion is exactly what it sounds like. It's where part of the DNA is just inverted, it's flipped over and swapped. And a way that we think this happens is actually when the DNA loops over itself and breaks right at the part where it crosses over, and then recombines back into the loop with that little section inverted. So this is how we think that most inversion mutations are happening right now. And the reason that we're sort of interested in these inversion mutations is that they can teach us other things about genetics. We looked at some really interesting studies looking at cryophages and proposing that there could be some sort of modular genetics happening. Modular means that there are a lot of different tiny parts that you can combine into a whole in different ways, which makes a lot of sense with phage mosaicism as well. And we were looking at a study that was looking into cryophage, which are phage that live at the very bottom of the ocean, very cold places. And they found a couple of different homologous genes that they believe are interchangeable. And one of these was the tail protein, which is where we saw our own inversions. So this is just interesting research that proposes that these inversions could mean something greater for phage genetics and genetics in general. So our conclusions are that these bacterial alignments ahead of the integrase genes suggest that this is the possible site of recombination, or the @P site. And these possible sequence inversions seem to be unique to our cluster Q minor tail proteins, which makes them an interesting way to study other genetics. So far, our research has been only genetics and no physical experiments, but if we were to test this further, we would try removing that @P site to see if the phage can form a lysogen, and we would also genetically replace the cluster Q minor tail protein with a different minor tail protein, just to see how these inversions affect the phage at large. And here are our acknowledgements. If you have any questions, we can take those now.
I saw some unusual names on a couple slides ago. Is that referring to proteins, or like how do you get names like Annihilator and Hotshot Baby 7?
Yeah, so those are actually each individual phages. So just like how, like if you took Phage Lab 1, you got to name your own phage. These are just how other people named them. So yeah, like Hotshot Baby 7. So yeah, these dot plots compare the entire genome of, so like Hotshot Baby against Annihilator.
Is there any like restrictions on the naming convention? It doesn't seem so.
I mean, like, I don't think there are some things I don't think you can name it like someone's full name, and definitely not something like super vulgar, but like Annihilator is kind of on the edge, but that's what it does to the bacteria.
Thank you.